Good afternoon, and thank you very much for the kind invitation. It's an honor for me to speak to you. My name is Torsten Haferlach, and I am the CEO of the Munich Leukemia Laboratory, and therefore I have to disclose that I am also part owner of the MLL. I would like to take the opportunity to talk to you about hematological diagnostics today and in the future, addressing aspects of today's gold standards, digitalization, automation, and artificial intelligence. Now let's talk about gold standards. This picture is more or less 55 years ago. At that time, the microscope looked quite different to that one I use today. However, at least if I look here, it seems that I started very early to be interested in looking into microscopes and looking into phenotypes of cells. Now, during the last 20 years, the WHO classification to which we have to adhere in hematology has been revised 3 times. And if you look on this cover here for a book of 500 pages and 5,000 references, there's still a lot of macroscopy and microscopy, but there is already some movement into genetically defined entities in AML or MDS, MPN or ALL. So starting with the phenotype of morphology, these are 2 very striking examples of an acute primary cytic leukemia on the left and the variant form of an APL on the right side, both bearing the translocation 1517, the PML-RARA fusion gene. And as you know, this diagnosis is very important and has to be given in a very short turnaround time because those patients can be cured in a cure rate of above 90% with the combination of all-trans retinoic acid and arsenic trioxide. Now, also this is somehow a pattern, a phenotype with flow. Right now we apply 10 colors. We are just on the way to implement 16 colors per vial, and the techniques today will also allow us to go for 21 different fluorescence dyes in one vial in the next years. However, this is the most important phenotype I would like to address today, in addition to molecular genetics, chromosome banding analysis with the viable cells. My wife is the head of this part of our laboratory, so this, I have to admit, I have never learned, but I completely know that this is a technique that needs a lot of experience on the wet lab and on the digital side of karyotyping. We also have more than 180 different fish probes for hematological samples here with double color, double fusion signals as shown here for 4 different fusion genes in AML. And we face around 15 to 20%, for example, in AML cases with a so-called complex aberrant karyotype with 3 or more chromosomes involved. And for this, we use for final solution and before we put down the ISCN formula, a technique called 24-color fish, where we can go on the same metaphase after gene banding and bring this to the final result of chromosome banding analysis. Now, the most important and very much growing part of the lab is molecular genetics. This is the hallmark of the CML BCR-ABL1 fusion gene, the translocation 922. This is not only true because we see more and more samples here at diagnosis, but a sharp increase of samples for follow-up since TKI was introduced into very successful treatment of those patients that now have a normal lifespan when treated with TKI after diagnosis of CML. And therefore, we run a lot of assays for MRD, measurable residual disease, with real-time PCR to detect the respective fusion gene and directly influence the treatment accordingly. So the composition of all these gold standards today is cytomorphology, cytogenetics, fish, molecular genetics, and immunophenotyping. We do not investigate refined biopsies here. This is done by pathologists in Germany, and we are hematologists and cytogeneticists, molecular geneticists. But you see here that there's a huge combination of these different techniques per case. 5 options there would be, and it's 2.5 per case in median that we have to combine to bring the final result to patients and doctors after we investigated those cases. Now let's move over to digitalization. We are all aware that there is barcoding all around in the world, in the supermarket, on our flight tickets, in our cell phones. We also have different other scanners that can read barcodes, and we have a lot of tablets all over the lab that hold the respective SOPs, any kind of assay and pipetting, and the complete LIMS system from the lab, so you're absolutely free to move around and don't need to run back to your desk computer all the time to see something or to add something to the database. I would like now to move over to automation, and we have a lot of in-house built-up solutions to automate our workflows in different parts of the lab. Now this is our way to analyze molecular genetics, and we start here with the lysis. We skipped FICO some years ago, and these robots help us with our peripheral blood and bone marrow aspirates to start with the molecular processing. And here you see the robot system we developed together with Hamilton and works with barcodes, starts here with the vials with the respective peripheral blood or bone marrow. And during several steps that are combined and connected by this H motion arm, on the left side there's a centrifuge that just opens. This robot processes the complete peripheral blood and bone marrow in a time of 2 hours from the respective sample to 2D barcoded little vials that we can finally cap and decap and then freeze in our freezers. This is how it looks like. These are the bottoms of the 96 well plate barcode system, and you have the caps and the decaps over there machine. And these are the freezers. The one on the front is -20 degrees centigrade, and the other one in the back of the room is -80 degrees centigrade, holding together more than 3 million vials. And on this machines, we start with this respective vials of 5 million cells in RLT buffer for later DNA and RNA preparation. This is the capping and the decapping of this 96 well plate format. And after this had happened, we move over to a scanning system like this here that now scans the respective barcodes. And from this moment on, these 96 well plates can move to the freezer, as you can see here. And in a system of so-called chaotic order, the freezers completely automatically organize the respective samples to go in, or for the next morning to go back to another 96 well plate for DNA and RNA preparation, including also the primers for the respective assays that the freezer holds in addition and places to the respective plates before they go for DNA and RNA preparation. So these systems are completely organized in our workflow and combined with our LIMS system. Now this is how we go for library preparation. We use several libraries for myeloid panels with 72 genes, lymphatic panel, and erythrocytosis panel as well, and we pipette into every respective sample a so-called human ID panel with 23 loci spanning the genomes with an incidence of 1 to 80,000 in humans to control the respective case when it is sent for the next time for any problem in pre-analytic procedure and, for example, also after allogeneic transplant. We use 2 MySics, and in addition we have 5 NovaSics to run all our panels between 50 and 100 a day, as well as our research and other applications, including genomes and transcriptomes and exomes. Why is this possible? Because on these respective machines and the respective flow cells, one can run up to 250 exomes per flow cell S4 in one run if you cover them 90x. If you go for genomes and cover 30x, you go for 24 per run. We cover our genomes around 100x, so we can implement 9 genomes per run, and the sequencing time is below 2 days with 44 hours. We then combine all these respective instruments from cell lysis, DNA extraction, library preparation, and finally sequencing and the storage freezers in between in a so-called Internet of Things that is completely organized by our laboratory information system, and also adding the information after sequencing to a completely automated bioinformatic pipeline to finally bring this information to our molecular biologists and cytogeneticists for. Finally, I would like to address the tools that we implemented in our lab over the last years with the support of AI and also accredited these tools. And I would like to start with cytogenetics, especially chromosome banding analysis. Now what you can see here is a metaphase captured by a slide feeder or metaphase finder from Metasystems. We worked together with this company since 1992 and bought our first metaphase finder in 1996. And what you can see here is a metaphase that is karyotyped by one of our most experienced technicians. Claudia Haferlach, my wife, is head of this part of our laboratory for cytogenetics and fish. And after separating the respective chromosomes here on a digital manner and ordering the chromosomes according to their size, they are usually by mouse click on the respective position in the karyogram. And as you can see here, it needs a lot of experience, phenotypical knowledge, and takes some time for starting with the metaphase after the metaphase finder to finally lead to a karyogram. And we need 20 metaphases for writing a report according to ISCN for hematological cases. And finally, this patient, who is a man and has a trisomy 8, is karyotyped. As you see here, that was a real-time movie, and you see that takes around 40 to 50 seconds per metaphase. So what we did, and Claudia investigated over the last years with her team and now use in routine, is the same metaphase as shown here. And now we start, and you see the support of AI, and the AI will help you to come to a karyogram like this. And then the human being has to control the respective karyogram before writing the report. But as you clearly have seen, this is quite quick and much quicker than the standard way to do it. And in addition, we now built up several additional tools that can use the metaphases when they are already captured by the metaphase finder and automatically karyotype them to give over the final karyograms for control of our employees. And here we go for 20 metaphases supported by AI automatically. And this new application can karyotype 1000 metaphases per hour to hand them over in the karyogram for control done by our technicians and cytogeneticists and biologists. So I think it's quite clear how supportive AI can be in a part of the lab like this. We also add this technique to the 24-color fish procedure, where we have the complex karyotype on the left side after G banding, and on the right side the same metaphase the next day after 24-color fish pipetting. And what you can see here, first on the left side, is the use of AI to go for a karyogram ending up with some marker chromosomes that you cannot put to the right position. Then the same metaphase next day, again, the use of AI. Here is solution given by the AI. And finally, you just transform the information from the right side to the left side, and you are done with this karyogram of a complex karyotype. Next, I would like to present some of our AI applications that we use in molecular genetics, especially for variant interpretation. As I have told you before, we start with this number of genes on our myeloid panel for sequencing, and this is the lymphatic panel with some overlaps, and after DNA extraction and quality control and library preparation with the respective panels, the human ID, as I explained to you before, we also pipette in all the samples CNV backbone spanning the whole genome, and after sequencing and quality controls, we go over to data interpretation and finally reporting. All that can be done within 2 days or even less. Just for some examples for CNV Backbone Panel, you very nicely can see here the gains of chromosome 12, 18, and 19 with a 50x coverage, and normally we cover our panels 1800x and minimum 400. This is only 50x coverage, and this is another case with the monosomy 7, but also a loss of heterozygosity in chromosome 12, leading to a deletion. So this is quite nice to see this just by using it in addition to our panel sequencing for NGS results, and also to help any cytogenetic result in parallel, especially for those cases with the very low rate of aberrations or when there are no viable cells in the cultures. So the tool that we use by AI support in molecular genetics is a random forest model with machine learning, and you see a specific variant here in TP53, and all these predictions tools are automatically organized by some AI-based algorithms and lead to an MLL predictor of 0.997, clearly demonstrating that this is a pathogenic or damaging mutation. And all this is based on our experience of more than 12 years of NGS in our lab with many hundred thousands of genes being sequenced. And another case, just another example, is this finding with another TP53 mutation where the predictor comes up with the score of 0.066. So this we would not vote for somatic or pathogenic. And this all leads to the workflow for data interpretation with data pre-processing as a first step, variant discovery, including several callers as a second step, and especially the variant interpretation with the use of worldwide available data sources such as Cosmic, ClinVar, and others, our huge in-house database over the last 12 years of NGS experience, and especially the MLL predictor, leading to a final screen that looks like that, where all the information of a respective finding that is available in Cosmic, ClinVar, DBSNP, and other databases, and our MLL predictor is given to our molecular biologists and bioinformaticians for final reporting, including the information if we saw this variant before, and if so, what was the respective final diagnosis of the patient, as shown here on the right button. Now, this has been organized and calculated all in our in-house local infrastructure some years ago with 15 minutes for runtime of all the bioinformatic pipelines per sample. But if we had 80 samples per day, it took us 80 times 15 minutes. So 2 years ago, we decided to move over to a own used cloud infrastructure. And there we hand over the respective information after sequencing, and then the different bioinformatic pipelines are run automatically in the cloud, and one sample with a lot of information in pipeline takes 40 minutes. However, if we have 80 samples, we just can process them in parallel. So it's also 40 minutes. If it would be 800 samples, again, it would be 40 minutes. And after calculation with all these respective pipelines, the final data interpretation goes back to our in-house storage system and is located in our laboratory. Now, the question is, will this be all fusion genes, some mutations of some specific panel genes, or will we move over to exomes, genomes, and transcriptomes in the future? And the even bigger question is, what will remain from gold standard diagnostics on the left side if we have available whole exome sequencing, whole genome sequencing, or whole transcriptome sequencing in the future? To understand this, and to make this possible, we first of all have to streamline the pipeline for genome sequencing, and we did this over the last years, and also finally accredited this pipeline, including the data processing in the cloud, taking 7 hours for a complete genome 100x covered. That finally leads to a turnaround time 7 days or even less for the complete process to write the final report. We tested this for more than 5,000 cases in our 5,000 genome project, as you can see here, with a lot of different entities of leukemias and lymphomas, nearly 800 AML, more than 700 MDS, and many other entities. And the beauty of this data set is not only that we cover 103x for the genome in median, but we also have transcriptomic data, including DNA-based data for 93% of patients. So this data set is now very rich for any kind of research, as well as for our support of any future routine diagnostic applications. This leads to a software that we program with our bioinformatic team, including every option for structural variant detection, automatically copy number variations, as you see here, with gains and losses spanning the complete DNA, and small nucleotide variants, and every other quality control marker is also included in this database, summing up finally to a report like that we already sent out starting 4 weeks from this day. So this is now one of our options for routine and leukemias and lymphomas for very difficult cases. And here you see the final report as a circuit plot with gains and losses, fusions like this translocation 1114, and the variants as shown here with the BRAF mutations on chromosome 7. We also use this huge data set of genomes and transcriptomes to test machine learning tools together with AWS. And here on the left, you see the true leukemia subtype as diagnosed by our gold standard diagnostic tools. And here the predicted leukemia subtype based on machine learning and the respective information that is located in the genome and the transcriptome. It's not all one in these boxes here, but it's quite understandable that if we see AML is 0.92, and some of the AMLs are classified as MDS or CMML, we know from our gold standard thresholds and experience that this is quite a neighbor of AML, and therefore we will improve this technique on one hand and also challenge the gold standards on the other hand by using such tools. This finally will be implemented in a cloud-based AI-driven infrastructure that we intend to get for FDA approval as a medical product, where any kind of information from the differential count of the blood to cytogenetics and metaphases, flow data and panels and genomes, transcriptomes, and also exomes could be uploaded by the respective user worldwide. The AI tools will process the respective data and give back the information, may it be on prediction, may it be on diagnosis or classification to the user after some seconds of cloud computing. Is this all far away? Well, this paper was published beginning of March this year from the colleagues from the Washington University, St. Louis, and they demonstrated in this paper that goes for genome sequencing as an alternative to cytogenetic analysis in myeloid cancer. And also they were able to run these assays in parallel to gold standards in a time frame below 1 week. and demonstrated that this is really very helpful tool, especially also for those cases where cytogenetics have problems with viable cells. Now, we refer to this paper in a correspondence and demonstrated that if we look into our 430 de novo AML, for whom we have panels with a coverage of 1,500, we have our genome with 100, and we can go for an in silico genome of 60x, that any kind of variant allele frequency in the range of 30, 25, 20, 15, or even 10 will lead to the same detection of variants found. However, if we go below a variant allele frequency of 10%, then we will miss, in comparison to the targeted panel sequencing with a very high coverage, some information in the 100x as well as even more in the 60x coverage of a genome, and 60x was used by the colleagues of the Washington University. So there is some discussion still needed what will be the final coverage for a genome. Now, all over, we think this will lead to the gold standards plus minus the tumor profiles or even the germline, maybe on a genomic level, leading to specific precision medicine drugs and offering some RMRD markers for control after treatment. This will not be the end. We also would like to know about prevention, because we have so many information right now in the system. We have information on the tumor as well, and then we want to participate and ask patients to be involved in those studies for prevention, for personalized medicine, and also for prediction, the P4 in cancer medicine. Because we hopefully are here at the stage of wellness, but something is going wrong, and normally this is the typical way that the disease is moving forward, and then the clinical diagnosis can be made. Now, we would be interested in the near future to see if we can detect first changes and then try to intervene by targeted therapy to get back and return to wellness. Eric Topol also put all this together in a very nice paper in the Nature Medicine 2019, where he added all the different information, including family history, tracking devices, microbiome, and all the genomic and transcriptomic information, and the world's literature, and put all this in a machine learning tool with artificial intelligence, leading to really health guidance and the respective patient's treatment. And he compared procedure like that with self-driving cars level between 0 and 5. Right now we here with nurses, doctors, and the patient's care, and we will move over step by step. I don't know how far it will be to be supported by any kind of data and AI-driven tools for treating the patients better, because Eric Topol concluded AI will not replace physicians. However, physicians who use AI will replace those who don't. And I just can recommend this very nice book, because at the end, if we go that way and we do a good job, then we may have more time to talk to our patients, and the patient will see us now again, and not we only are sitting at our desk and controlling data sets and looking for papers that we are missing with reports and results. So finally, I would like to thank my team, especially Claudia, for all her great job in cytogenetics and FISH and AI implementation in our daily routine. Wolfgang Kern in Flow. Manja Meckendorfer, she is the head of our molecular department, including research and development, and also has driven and is driving the 5,000 genome study. And Wenke, Niroshan, and Stefan, three of our bioinformaticians who are really very fascinated in working with us in this field, and our complete team of doctors, cytogeneticists, and molecular biologists, as well as technicians, nurses, and so on. They work with us in our gold standard approaches every day for our patients, but are open for any use and testing of AI tools for today and in the next future. Thank you very much.